In this video, we learn more about regulation at the post-transcriptional and translational levels. Once an mRNA is made, a series of events must occur if the final product is going to affect the cell. Each of these events offers an opportunity to regulate gene expression. The four major control points at this stage include splicing mRNAs in various ways, modifying the lifespan of mRNAs, altering the rate at which translation is initiated, and modulating protein function after translation is complete. We'll look at each of these mechanisms in more detail. Introns are spliced out of primary RNA transcripts while they are still in the nucleus. Alternative splicing leads to the production of slightly different mRNAs that then lead to the synthesis of different proteins inside the cell. Changes in gene expression are possible because selected exons may be removed from the primary transcript along with the introns. As a result, the same primary RNA transcript can yield more than one kind of mature processed mRNA consisting of different combinations of transcribed exons. If we look at the gene for tropomyosin, a protein present inside muscle cells, we see that it contains several exons as well as introns that will later be removed from the mRNA transcript. Once this primary mRNA transcript is made, it can be processed in multiple ways, leading to the removal of certain exons in addition to introns, generating slightly different mRNA transcripts that will lead to the synthesis of slightly different tropomyosin proteins depending upon whether it is a skeletal muscle cell or a smooth muscle cell. This makes sense because the skeletal muscle cells are involved in facilitating movement of bones and joints, whereas smooth muscle cells surround all the internal organs inside our body and help in small-scale contractions, for example, those in the small intestine to move food along the digestive tract. Alternative splicing is regulated by proteins that bind to the mRNAs in the nucleus. These proteins interact with the spliceosomes, which are complexes that catalyze the splicing of mRNAs. Over 90% of human sequences for primary mRNA transcripts undergo this process of alternative splicing. Although humans have just 20,500 genes, we can produce at least 50,000 proteins. This is more than twice the number of genes that we have. And alternative splicing is one of the mechanisms that allows this diverse population of proteins to be made within the cell. Based on these findings, genes are now considered to be the coding and regulatory sequences that direct the production of one or more related proteins or RNAs. The second mechanism that is involved in regulating gene expression after transcription is complete relates to mRNA stability. Once the spliced mRNA leaves the nucleus, in the cytoplasm, additional regulatory mechanisms control gene expression. As a result, stability of mRNAs in the cytoplasm is highly variable. Some mRNAs are degraded rapidly, allowing for only a short period of translation, whereas others are quite stable, leading to extended periods of translation. A process called RNA interference controls the lifespan of mRNA in many cases. During this process, tiny single-stranded RNA molecules called microRNAs or miRNAs are made which target specific mRNAs. These microRNAs bind to proteins that are part of a complex called the RISC protein complex, after which, in combination with the RISC complex, miRNAs bind to complementary sequences in the mRNA. When part of an mRNA becomes double-stranded, this sends a signal to specific proteins to degrade that mRNA or prevent it from being translated into a polypeptide. About 20 to 23% of all animal and plant genes are regulated by miRNAs. As we can see in this image, microRNAs are also transcribed from genes that are present within the DNA of an organism, leading to the formation of a precursor hairpin loop structure that is then exported out of the nucleus and processed to form an miRNA duplex, which is double-stranded. The two strands in this duplex then separate, and the single-stranded mature miRNA binds the RISC protein complex before it can start targeting mRNAs. In combination with the RISC complex, the mature microRNA then binds to a complementary target mRNA sequence, which results in the degradation of the target mRNA by the enzymatically active part of the RISC complex. Once the mRNA is degraded, it cannot be translated to form proteins, and this is how gene expression is regulated through RNA interference. The third mechanism that helps regulate gene expression is the control of translation itself. These mechanisms are responsible for controlling the timing or rate of translation. For example, in response to viral infection, translation can be slowed or stopped by phosphorylation of a certain ribosomal protein. This is a quick response to situations in which cells are in danger. Other situations include exposure to sudden changes in temperature that lead to modification of the translational machinery, reducing the rate of translation. The fourth and final mechanism for regulation of gene expression occurs at the post-translational level. After translation is complete, these mechanisms allow the cell to respond to new conditions rapidly by activating or inactivating existing proteins. This means that the cell already invests a lot of energy in using the information in DNA to form RNA and to build proteins that are ready and waiting to carry out functions within the cell on the receipt of a signal from the outside, for example, a change in the environment of the cell. This saves times for the cell because these proteins are ready to go. But there's a trade-off because the cell has already invested a lot of resources in building these protein molecules that may or may not be required in the amounts in which they are made and might later be degraded without being used. Some other mechanisms that lead to post-translational control include the activity of chaperone proteins that regulate protein folding in the appropriate conformation conducive to their function. There are enzymes that modify proteins by adding carbohydrate groups or cutting off certain amino acids. This leads to the activation of those proteins. A common example of this is the enzyme lactase, which helps digest the milk sugar lactose. Another mechanism for regulating protein function is phosphorylation, or the addition of phosphate groups on proteins. Phosphorylation can either activate or deactivate proteins, depending on the nature of the target protein. The activity of already made proteins can also be regulated by destroying them through the process of tagging with ubiquitin tags. This prevents proteins from performing a role when they are not needed. These ubiquitin tags are recognized by a multimolecular machine called a proteasome, which cuts the proteins into short segments, which can then be reused or recycled to build new proteins. So the proteasome acts as a sort of waste processing unit that keeps digesting proteins that are not needed inside the cell any longer. This concludes the section on regulation of translation. After going through this information, I'd like you to be able to respond to the questions listed on this slide. Thank you.